So in the lab, the thing that I like the most doing would have to be the protein assays, because even though all of my coworkers really hate it because they find it really tedious and no one wants to do it and they've done like 50 of them, for me it's all new. So every chance I get to do one, I just go for it. In my opinion, the most interesting tour would have to be the one where we went to the nanofab facility, because I got to learn a lot about like electron microscopes and about particles and just what kind of equipment they work with. And I also got to meet this chemistry teacher. So I'm hoping that maybe I'll go into chemistry later in life and he might be able to answer a few of my questions.
The most complicated phrase I've heard at HYRS 2014 is mTORC1, mammalian target of rapamycin complex 1. One of the most interesting things I've learned about researchers at the U of A is that things take a long time to do and you have to repeat them numerous times to prove your results, and oftentimes they're very frustrating, but you have to keep persevering with it.
Well, the most interesting thing for me was looking at stem cells. Stem cells have the ability to differentiate into anything almost, and they do this by silencing certain genes. My favorite social activity this summer was the HYRS Olympics. So we all went to the park and we played soccer, but in the end it turned out into a huge water balloon fight, which was super fun.
So far, the most interesting thing I've learned in my lab is about SH and siRNA. SH meaning short hairpin RNA and Si meaning small interfering RNA. They're two special types of RNA that interfere with the protein synthesis process and prevent a protein from being synthesized. So far, I'm looking forward to the brown bag lunch, where we're going to be looking at microscopy with a scanning electron microscope.
The most interesting thing that I've seen is probably perfusion, because I got to see someone cut up a mouse and then they took the heart out and then they put it onto a pin and it kept on beating for about an hour or so. That was really interesting because they kept it alive. My favorite tour was the IRSM, because the IRSM found new ways to help improve science using technology, which helped people with head and neck cancer.
The most interesting brown bag lunch that I attended at HYRS was the one where we got to meet people that had gone and volunteered abroad to places like Africa, and I thought that was a really interesting brown bag lunch because it showed me, where I could go and what I could do when I got older, go travel and help people in other countries, so that was really interesting. I think one of my favorite parts about being in the lab is when I got to go down to SMRI and I got to perform a pig surgery. I got to isolate the spleen and the lymph node cells to use in a future experiment, so got to learn proper surgical procedures, so that was really neat.
This summer, the most interesting thing I learned was the relationship between the gut microbiota and the immune system, so how your diet can impact the way that these bacteria and the cells interact with each other, and that can have a big impact on your immune system's ability to recover yourself. My favorite social activity, I liked all of them because they were a really good opportunity for all of the HYRS kids to bond and get to know each other better. I especially liked the when we played in the park together and we just had a water fight and we relaxed and we had a potluck and stuff, so that was really enjoyable.
My favorite brown bag lunch so far this summer was when former HYRS students came and talked about their experience volunteering overseas in Africa and in South America, and I thought that was a really cool thing for them to tell us because we would also have the opportunity to go there if we wanted to. My favorite tour so far this summer was when we went to the Misericordia Hospital and we went to our IRSM, and we got to see people who worked with computers, but they're making bones on computers, and then they got to 3D print them, and then they could be surgically inserted into people's bodies, which I thought was really sweet. So yeah.